Hey everyone, in this video we're going to talk about different media that we use, and we may, depending on the time, finish up talking about how we can determine the amount of growth that is occurring or the amount of organisms in any given sample. So when we're growing microbes in our lab, there's a lot of different types of media that we can use. Many of the media that we use are called complex media. These are going to contain many different ingredients that grow and support a variety of organisms. Typically, these organisms are non-fastidious, so they can grow without any special nutrients being available. Sometimes we use chemically defined media. This is going to be used mainly in labs where we need to grow organisms, specific types of organisms, maybe fastidious organisms, or we're trying to control the growth of a specific type of E. coli strain. Okay. So over here you can see some different agars that are used, or not all of these are used in our microbiology lab, but some of them are used in microbiology labs. In our microbiology labs, I should say. So a complex medium versus a chemically defined medium. Complex is going to just have a list of basic items. So there's water, beef extract, and peptone. But there's no exact amount of anything, and we don't know what's in the beef extract. Whereas chemically defined is going to have every single item defined in it. There's iron sulfate, calcium chloride, ammonium sulfate. That tells you how much of each and what types of materials are in there. So our complex media grows all organisms. Selective media is going to grow only certain types of organisms that we care about, and it inhibits growth of other organisms. So in selective media, we can grow certain species from a sample, like you might want to grow only gram positives or only gram negatives, while allowing other organisms so allowing certain organisms to grow while inhibiting the growth of other organisms. One example of a selective media is McConkey agar. It is selective for gram-negative bacteria. It contains crystal violet. Crystal violet inhibits gram-positive growth. It contains bile salts that inhibit most non-intestinal bacteria. And so a lot of our gram-negatives live in our intestines. So these are enteric organisms, or I should say our enteric organisms live in our gut, and most of them are gram-negatives. But there are some gram-positives, and the crystal violet inhibits those guys. Differential media contains substances that separate organisms based on how they look when they grow. And so here we have blood agar. Blood agar produces different characteristics depending on what organism you're plating. So here we have no hemolysis. So this is like what we would call gamma hemolysis. Gamma hemolysis means there's no breaking down of blood. Here we have alpha hemolysis. Alpha hemolysis is partial breakdown of blood. And then beta hemolysis, this is full breakdown of blood. Beta hemolytic bacteria are the ones that can cause things like strep throat. Alpha hemolytic bacteria can cause disorders, but probably not as severe as beta hemolytic. But again, they all grew. It just separated them based on how they grew. So you can have a complex media that grows everything. And so here we have 5 different types of organisms growing. This is just a typical medium, right? A routine medium like not fluid thioglycan, I'm sorry, like tryptic soy agar or nutrient agar. Selective media is going to select for certain organisms to grow and other organisms don't grow. So in this case, B and D did not grow, whereas ACE did. Differential media separate organisms based on how they look. So these two organisms, E and B, look different from A, C, and D. And then if you have a selective and differential media together, that allows you to grow organisms, and only organisms that can grow will grow, ACE, but E looks different from A and C. So we can use enrichment cultures to isolate and promote the growth of specific organisms in a broth. So say we have a sample of pond water, we can actually provide the nutrients needed for a specific organism to grow very effectively. And then we can grow them and make them the most common organism in the sample. This allows us then to pull them off and produce eventually a pure culture of that organism. So the last thing we're going to go through is how we can detect growth of microbes. There are multiple ways that we can detect growth. I'm going to quickly go through these. One way, this is relatively simple to but it doesn't only detect living cells, is direct cell count. So this counts the number of cells that are living and the number of cells that are dead using a flow cytometer. And so you can see you use the special type of slide that you put the fluid in, and then it has a grid, and you just count the number of cells within each section of a grid. Viable cell count is going to be counting only cells that can actually grow. And so we use a selective and differential media if we're going to want specific types of organisms to grow. Otherwise, we just use a complex media to see what all organisms might grow. And using viable cell count, we are going to then take and continue to dilute a sample multiple times until we have about a 1 in 10 dilution. We then plate the bacteria, and the idea is the initial plate is going to be very, very, very dense with bacteria. But every time we dilute it, we should lower the number of cells. And eventually at that 1 to 10 dilution, we should have only about a few cells in there that are going to grow, and so we can count the actual colonies. So here you have 1 millimeter of a culture to 9 milliliters of some broth solution. Then we take from this, and we take 1 milliliter of this sample, add it to 9 milliliters of another broth that's sterile. We then take this and we add it to another broth that is sterile. And we keep doing this until we have about a 1 in 10,000 dilution. So the 1 in 10 dilution step takes multiple. So you're diluting it each time 1 milliliter to 9 milliliters of some solution. And eventually you should have very few cells. You plate them, and here you're going to be able to see the number of cells. Here you hopefully will have between 30 and 300 cells. A spread plate method or pour plate method helps us to identify only colony forming units. So in this case, cells are often attached to one another, or in this case, what you do is you're going to take a solution that has been diluted and you then spread it around. So here we have the dilutions. We take this dilutant and we can put bacteria on here and then use a glass spreader to spread the solution all the way around. Or we can pour it into a melted agar and then the bacteria will grow either on or below the surface. This one will allow you to grow anaerobic organisms. This will only grow the aerobic organisms. Membrane filtration allows you to find microbes that are in a solution as you are filtering that solution through a specific membrane. It's only going to filter microbes that are too big to get through that filter. And then you can take the filter and you can grow it onto an agar plate, and then you can count the number of colonies, and then you can see how many actual organisms were there. Most probable number is an estimate. So this is going to be an estimate of a larger number. You're going to take multiple tubes, incubate them, and record the number of organisms based on the amount of gas or the amount of byproducts produced. So here we have 4 gas bubbles, and so with that you're going to look over here, combination of positives. We have 4, 1, 2, 3, 4, and then you have in the next one 3, and then in this one 1. So we're looking at 4, 3, 1. So the most probable number is there's about 33 in a 100 milliliter solution. That's how you would figure out most probable number. They have these tables on the back of most of our microbiology textbooks and lab books. We can also measure the biomass. So we can do this by measuring turbidity using a spectrophotometer. So what this does is it sends light into a solution. If the solution is very turbid, the light is going to be bounced back. If the solution is very clear, the light will go all the way through. So here's a non-turbid and here's a turbid solution. Here you can see light moving through and hitting because it's not turbid. Here you see a lot of bouncing of light, and that shows much less absorbance, so that's more turbid. So here's just your table showing all the different ways that we can identify growth of microbes. And this is it for this class or for this chapter. So I'm going to end this video and I'll post all these videos so you can watch them. Bye.